Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall talk about the extrinsic pathway of apoptosis using the receptor Fas-R or trail R, which is also very similar to Fas-R. So in the last video, we talked about extrinsic apoptosis using the TNFR1 receptor, where the ligand is tumor necrosis factor alpha. In today's video, we shall look at the Fas receptor, which is very similar to the trail R1 receptors or the trail R2 receptor. So we'll just focus on the Fas receptor. So this is what we'll talk about today. The Fas-R receptor is a transmembrane protein. This is found in most cells. It has an extracellular cysteine-rich domain, CRD, cysteine-rich domain, and this is the domain, the extracellular domain, that binds the ligand, the Fas ligand. Then it also has a transmembrane domain that allows the receptor to be embedded in the plasma membrane. And it has a DET domain, a DD domain, intracellular DET domain. So this is basically what this receptor is. It has an extracellular, transmembrane, and an intracellular component. The ligand that binds this receptor is called the Fas ligand, sometimes called FASLG. This is a transmembrane protein found on the plasma membrane of certain cell types, especially predominant in activated T cells, such as the cytotoxic T lymphocytes or CTLs, as well as the natural killer cells or NK cells. Similar to what we saw in the last video on the TNFR1 signaling, the Fas-R signaling can also trigger either cell death by extrinsic apoptosis or necroptosis, or it can trigger a pro-survival, pro-inflammatory program. So very similar to what we saw last video, we have three possibilities of the Fas-R or the trail R receptor signaling, either cell death or cell survival. Let's first discuss how the Fas-R triggers cell death by extrinsic apoptosis. So we'll look at this part first, and then we'll talk about the remaining two, necroptosis and the pro-survival program. So as before, the first step is the ligand binding to the receptor. So the Fas-L, which is the ligand, will bind the cysteine-rich domain of the Fas receptor, and these receptors will be present on the plasma membrane of the cell that is going to either die by apoptosis or necroptosis. For example, in a cell that is infected with viruses or some other intracellular pathogens, a cancer cell, or a cell that is no longer required. The binding of Fas ligand to the receptor, to the Fas-R, causes the exposure of the DET domain of Fas-R. So this binding exposes the DET domain. On a side note, it's important as a healthcare professional to appreciate this. Antibodies that resemble or mimic signals such as Fas-L can also sometimes trigger their receptors. They will bind. So even antibodies that resemble the signal, they can bind receptors and trigger the cell signaling pathway. So this is an important concept to understand because there are several drugs that have been developed or are being developed that actually are antibodies that are used to trigger receptors, and there's diseases where antibodies are produced in the body and they resemble the signal and that can trigger receptor activation. So this is a side note but an important concept to understand in general about receptors and cell signaling. So when the signal binds receptor, that domain of the receptor is exposed, and now that domain of the receptor, this DD of the receptor, will interact and bind with the DD of a protein we talked about in the last video, and that is Fas-associated dead domain, or FADD. So the DD of the Fas receptor will interact with the DD of FADD, and that will recruit FADD to this Fas receptor. Recall from the last video when we talked about TNFR1, we also saw the importance of this FADD. It is a predominantly cytoplasmic protein, but there is research showing that it may also be present in the nucleus. Okay, so this once again is involved here in case of the Fas receptor and what we saw previously with the TNFR1 receptor. Recent research shows that actually FADD is involved in many cellular processes besides cell death by apoptosis or necroptosis or even autophagy. It's involved in cell cycle, in tumor progression, and very important in inflammation. So sometimes it's considered as a master regulator of cell death and inflammatory pathways. So as a healthcare professional, very, very, very important to appreciate, understand this protein, and that is called FADD. So once this FADD is bound to the Fas receptor, that interaction also exposes another domain on FADD, and that's called the DD domain. The DD domain. And now this the exposed DD domain on FAD will bind the DD present on the pro-caspase 8 or 10. So this pro-caspase 8 or 10 also has DD domains. So there's an interaction between the DD domain of FAD. So this will interact with one of the DD domains on caspase 8 or 10. So once this DD domain binds with one of the DD domains of caspase 8, now that will lead to the exposure of the second DD domain on caspase 8. So this DD domain now will interact with another DD domain on another caspase 8 molecule and expose its DD, and this continues and forms that leads to a chain reaction and forms a pro-caspase 8 filament, basically made up of DD oligomers, and this is something we also saw in the previous video when we discussed the TNFR1 pathway. So now we have several pro-caspase 8 and 10 very close to one another. This increases its local concentration, and it forces these caspases coming together to form dimers. And once they form dimers, they become auto-activated and they can start cleaving each other to form active caspase 8-10 enzymes. So now we have active caspase 8-10 enzymes. Now this complex of the receptor, the FADD, as well as the caspases, here this is called the that inducing signaling complex, or DISC, which we also saw in the last video. So once this ligand binds to the receptor, this forms very quickly, within seconds. Now this active caspase 8 or 10 will then go on to cleave the executioner caspases, and then this executioner caspases will cleave selected proteins in the cell, leading to apoptosis, and we'll talk about these executioner caspases in a future video. So just within a few minutes of ligand binding to receptor, we see this apoptosis happening. And this is what we typically see in some cells, what we call type 1 cells, such as lymphocytes. Now in type 2 cells, for example, hepatocytes, this active caspase 8, besides cleaving the executioner caspases, they also have to do another thing. They will have to cleave the pro-apoptotic protein bit into T bit. And this T bit will now go to the mitochondria and activate the back assembly, and that leads to the permeabilization of the mitochondria, which then leads to intrinsic apoptosis that we talked about in a previous video. So in this case, there is extrinsic as well as intrinsic apoptosis that will kill the cell, and this is what we see in type 2 cells. So this basically is how the Fas receptor causes cell death by apoptosis. This process that we just described is a very important way by which the cytotoxic T lymphocytes or the NK cells are able to kill a cell that is infected with some sort of intracellular pathogen, such as a virus or some other intracellular pathogen. And this is also how these CTLs and NK cells can kill cancer cells. Okay, so this process we talked about, apoptosis, is a way by which these CTLs and NK cells kill these cancer cells or infected cells. There is another way, and that is they release these CTLs, NK cells release proteins called perforins and granzymes, and these perforins and granzymes can then go on to kill the infected cell or the cancer cell, and that's something we'll talk about in a future video under the biochemistry of the immune system. Now it's not surprising, since this Fas signaling will kill the cancer cell by apoptosis, that the cancer cells, certain cancer cells, what they do is they reduce the amount of Fas receptors on their plasma membrane. And by not having Fas receptors, or very small amount of Fas receptors, these cells can avoid cell death by apoptosis. So these cancer cells survive and keep on proliferating to increase the size of a tumor. Remember, signal receptor interaction is very statistical. So it really depends on the concentration of the receptor as well as the concentration of the signal. So by the cancer cells reducing the concentration of receptor, this interaction will decrease. And therefore the cancer cell will not undergo apoptosis, and it will survive and proliferate. As we discussed in the previous video, then we have proteins called FLIPs, which is produced by certain viruses and once again by certain cancer cells. And you can see that the enzyme caspase 810 uses its DD to bind to fat, to the DD of fat. But these FLIPs also have DDs. So they too can bind to fat. But FLIPs do not have caspase activity. Okay, so they simply bind and they prevent this caspase-8 from binding fat and becoming activated. So basically what they are doing is they are inhibiting caspase-8. Okay, so they are inhibiting caspase-8. So by flips inhibiting caspase-8 through this competitive inhibition, these executional caspases are not activated and apoptosis doesn't happen. So that's how some viruses and cancer cells survive, by stopping apoptosis, by secreting or producing the protein called FLIP. Okay, so that's how Fas-R causes extrinsic apoptosis. Now let's look at how this necroptosis program is triggered by Fas-R. Now, if for any reason caspase-8 is inhibited, now the cell is not going to go through apoptosis. What happens now is fat, this very important protein fat, will now recruit another protein, RIPK1. It will recruit RIPK1, which will then recruit RIPK3, which will then recruit MLKL, and this will now trigger cell death by necroptosis. This is something we'll talk about in a future video. So in any scenario where caspase-8 is inhibited, this cell can still die. In this case, it will die not by apoptosis, but by necroptosis. And as we saw in the previous video, since many viruses produce inhibitors of caspase-8, it is believed that necroptosis may have probably evolved in a human as an antiviral defense mechanism. So if the viruses are going to stop the cell from dying by apoptosis, so now the cell is going to say, okay, you are stopping me from dying by apoptosis and protecting the human. No worries, I have another program. I'm going to use necroptosis. Now let's see how the Fas-R signaling triggers the pro-survival, pro-inflammatory program. So if apoptosis doesn't happen and necroptosis doesn't happen, now the Fas receptor signaling can lead to the pro-survival program. And in this case, once again, fat plays a very important role. So fat will now recruit many of the proteins that we discussed in the last video, under TNFR1 signaling, complex 1 formation, and these proteins together with caspase-8 form what we call the phadosome. So this is a cytoplasmic phadosome that is formed, and in this phadosome caspase-8 is required, but not the catalytic, not the kinase activity of caspase-8. Just caspase-8, but not necessarily its activated form of caspase-8. So in this cytoplasmic phadosome, these proteins will, once again, similar to TNFR1, will stimulate this IKK and TAK1 protein, and that will trigger the NF-kappa B pathway as well as the MAPKK signaling pathway, and both of these will then cause or trigger the pro-survival, pro-inflammatory program, similar to what we talked about in the last video. So this is how the Fas-R or trail is very similar, so we can substitute Fas-R with trail. So this is how these Fas-R signaling causes extrinsic apoptosis, and if that doesn't happen, necroptosis, or it can promote the pro-survival, pro-inflammatory program. In the next video, we shall continue with extrinsic apoptosis, but we'll discuss a different receptors. That would be the TLR receptor as well as the ZBP receptor. So until the next video, thank you very much for watching. Have a wonderful day and we'll see you soon.